Just to make sure, Steve, are you still on?
Yep.
Okay, great. So we have the panel discussion, and first I would like to introduce my co-chair, Tom Curran, who is also a representative of the Forum on Drug Development, who actually helped also on the organization of the workshop. Thanks, Tom.
Thank you very much, Samir. I'm Tom Curran from Children's Mercy, Kansas City. First, want to thank all the speakers for really stimulating presentations. Want to move into the Q&A section. For those of you present, please use the microphone and give your affiliation and name when you ask a question. Those in the virtual world, please enter your questions into the chat, and Laurie will be asking them on your behalf. And let me kick off and ask the panel. We're very much interested in getting take-home messages. So what would you say, and each of you can answer this, are the key challenges and opportunities for single-agent immunotherapeutic development? Okay, I'm going to go to Steve first. And Steve, maybe I can ask you an additional one on top of that, which is, are there unique challenges in pediatrics?
Okay, I'll try to answer both those questions, and great to see you virtually, Tom. So as far as the key challenge in terms of single agent, obviously CAR-T has fairly significant single agent activities, obviously, again, in blood cancers and not in solid tumors. But we're seeing the limitations of that, and I think there is some impetus in the field to go to dual antigen targeting. I think there are opportunities for improving cell manufacturing that we're just beginning to touch, and that the next 5+ years, I think it's going to be a major part of the story. But in the end, for solid tumors, we still have the trafficking problem, whether you're looking at single agent or dual agent approaches. Getting the T cells in and having them not be inactivated is a major part of the problem. And some of that is a manufacturing issue, but a lot of that is going to be adding other pieces to the T cell engineering. So I think that figuring out trafficking for solid tumors is our major problem. And then in peds, we're no better off than the adults in terms of responses to solid tumors, and have fewer options in terms of targeted cell therapy. So figuring out targets is what the pediatric dream team through Stand Up to Cancer has done, led by Crystal McCall and John Maris. And I think that we're continuing to work on the targeting problem, but we still have the trafficking problem that I think is very similar in peds and adults. And of course, if you look at the broader world of checkpoint inhibitors and similar therapeutics, the lack of useful mutations in most pediatric tumors, which are quite cold, is another barrier for us.
Thank you, Steve. John, do you have a quick recommendation for us?
Yeah, I guess it all comes down to biomarkers. I mean, we have a number of agents that have limited activity at the 5 to 12, 15% range, and what it is about those tumors that allows them to respond to a single agent or potentially at a higher rate in combination is the fact that we just don't know how to identify those patients. And
Can I add something to John? John, just since you're, so from your discussion and from your presentation, again, the reason we're doing this is lesson learned. So from what you discussed, particularly regarding co-stims, OX40 or GITR, it showed two things. One is that you did not have any reason to feel or believe that it could work as a single agent.
yeah, that
And the second is, and I'll talk a little bit about that during the combination stuff, is that the combination showed that there is a reason not to use it in that particular combination. So my question to you is, retrospectively, and you worked on those compounds, whether in academia or in clinical, in industry, in biotechs, would you have taken those, or at least the OX40, into a single agent first, based on the information that you had, and what else you would have done to make sure that, yeah, that is worth testing?
Yeah, I mean, I think we obviously got way ahead of, there are two potential mechanisms. I think that, the T regulatory cell depletion, which is, I believe, the primary mechanism by which these antibodies work in animal models. The GITR antibodies perhaps modulate or destabilize the T regulatory cell population and turn them into Th9 effector cells in the models that do that. Our GITR antibody at Incyte didn't have that activity. But I think understanding what it is that you're going for, if you're trying to stimulate a population of T cells, perhaps we would have done better with a non-FC receptor binding or antibody than an IgG1, perhaps.
Okay, can I transition to Hussein? Because that's very much related. You also showed clearly that LAG-3 did not work as a single agent, and you showed very good mouse models that are probably fit to test this, whether MC38 or B16. And that's going to go to Suzanne afterward, because what's the criteria? So clearly, you did not show LAG-3 single agent. Again, would you have taken it as a single agent? Because this is one of those issues relevant to policy, is when do you really need to test it as single agent or just jump into combination in the first place?
Well, I really appreciated the way you guys set up this first session, and I love the fact that my title was single agent activity, which my answer is going to be doesn't work. And that's kind of, I think where we're getting here is that cancer immunotherapy is vast, and we go from cellular therapies to agonists to now checkpoints, and I think we just need to be very clear about what we're trying to achieve with a drug. If we're trying, again, if we know the mechanism and at least we know an inkling of why we're doing it, we have to be able to choose the right population, but also choose the right companion with it and the right combination. So expecting single agent activity at this point, I think, is not appropriate. I think being able to, we should be asking ourselves as drug developers and the FDA, what is our drug development strategy for this particular target? Not what is the single agent capable of doing. And so if that single agent, if that particular target requires two different things to be, kind of inhibited at the same time. We're kind of working now on doing CTLA-4 PD-1 and LAG-3, and I'd be interested if we augment TCR signaling that much, will the amplification of LAG-3 still matters? And if it always will add 25% over what single agent PD-1 and now the combinations, then we will have an even more effective combination. So we may be going into triplets, quadruplets. And the question goes back to what is the mechanism that we're trying to actually target and reverse, and which patient population is it most appropriate in?
So the last question here, and then we'll go to the floor. Suzanne, you showed drug that worked, 2 drugs that worked, and we've seen few drugs that did not work. And based on that, and clearly you've outlined the reason why PD-1 could have worked. 1, the concentration in tumor. 2nd, you had pretty good selection of patients, despite the fact that PD-L1 negative could also work. And maybe we did or we did not predict that. But can you come up with a criteria and say, look, this would work as a single agent. I know I'm simplifying it. This will never work on a single agent. And I know in between there are lots of gray area, but at least can we make that definitive for the extremes?
Yeah, unfortunately, I think the preclinical models can only take us so far, because there are a lot of things that do not translate literally from mouse models to human cancer. And in fact, anti-PD-1 activity in mice, I think it way underestimates what anti-PD-1 can do in humans. And there are a lot of technical reasons why that might be the case. So for me, we do need to have some clinical data testing these new drugs as single agents. And the big question is, what is the most efficient way to test single agent safety and activity and then quickly move on to combination if it turns out? What are the criteria to declare this is not working as a single agent, but it's still safe, and now we're going to move to combination. So there are, I mean, Hussein showed the LAG-3 trial design. I mean, that trial was designed over 10 years ago. So there were a lot of patients who got LAG-3 monotherapy in a dose escalation format. But could that have been done more efficiently if each patient had a brief run-in with LAG-3 monotherapy and then transitioned to combo? Or, there are many clinical trial design options. Maybe some people in the audience would want to comment on that. Thanks.
So let's go over here for the first question from the audience.
Amat Rahini, Moffitt Cancer Center. My question is Dr. Janik about the failure of the molecules targeting the positive co-signaling molecules. We just presented an observation at SITC last week. George Wiener is a co-author about the differences in co-expression between co-inhibitory and co-stimulatory molecules along with PD-1. And we found that tumors like melanoma and urothelial carcinoma as more immunogenic tumors, they tend to have a PD-1 plus immunoinhibitory dominant phenotype, while others like ovarian cancer, pancreatic cancer tend to have PD-1 plus immunostimulatory phenotype. So are we testing those so we're thinking that possibly these are tumors where we should be testing those, co-stimulatory molecules, and whether there is data in this area, in those ovarian, pancreatic, and so on.
Yeah, I mean, we collected a large number of tumor biopsies during our clinical trial in the pre and post state. And the results of those did not really help us for a single agent was where they were primarily examined, did not really help us to distinguish responsive from non-responsive tumors. There was only one patient in our GITR trial and one patient in the OX40 trial at Insight that demonstrated single agent activity, and this was in a population of about 100 patients each in those trials. So, I'm not sure how well we performed the immunohistochemistry to look at multiple, but that's obviously, we had only a limited panel of about 10 tumors that we looked at multiple markers in those patients. So that's always the challenge with the biopsy results, that there's typically very small samples, and the panel of assays that was performed is not probably adequate to really give us understanding. And of course, as is typical, the two patients in both of those trials who responded didn't have pre-treatment biopsies available to analyze. So
Let's switch over to George on this side.
Sure. Well, first of all, thank George Wiener from Iowa. Thank you for the great talks. I have what I know is a loaded question for Dr. Topalian. So there are obviously a number of anti-PD-1 antibodies in the clinic and in development. Do you think that they're mechanistically and biologically interchangeable? And from a scientific point of view, and from a societal point of view, should we be investing in additional anti-PD-1 antibodies, or should we really be focusing on understanding potential combinations?
Thanks, George, for that tough question. So I think for the anti-PD-1s, at least for the first three that went into the clinic, because we don't know as much about dostarlimab, but for the others, Nivo, Pembro, and Cemiplimab, I consider them to be interchangeable. I think we know enough about them, that even though they haven't been cross-compared in the same trial, to know that they have very similar mechanisms of action, very similar safety profiles, and very similar efficacy. It's not so clear with the anti-PD-L1 agents, and there is discussion now in non-pharma groups about designing trials to compare them head-on, because they do have different isotypes, and some of them fix complement or bind to Fc receptors, and others do not. There's a general perception that they may not be as active as the anti-PD-1s, but again, they have not been compared head-to-head.
If I might jump in, actually, I would also tell you that, kind of going to LAG-3, when relatlimab was initially an antibody that was designed to block the MHC class II interaction, and thankfully it turned out that it blocks FGL1 as well. But again, there's a lot that we're learning about that mechanism that I do expect different approaches to LAG-3 inhibition to actually target different binding companions, different ligands, and even different intracellular signaling. So I think as we learn more intricately about the biology of these checkpoints, we may actually start developing newer approaches to target them.
We did learn that with anti-CD20 antibodies, that when they bind differently to the ligand, they can have different effects. So that's consistent there. Thank you.
One other comment with regard to PD-1, John Wary has done a lot of work with regard to what happens to the T cells upon interaction with PD-1. And his studies demonstrate that about 70% of patients show a peripheral blood Ki-67 induction, and that Ki-67 induction occurs only once. And yet patients respond to the therapy. And so the big question is, are there agents or ways to administer PD-1 blockade in such a way that we can have that reactivation occur? So he was discussing at the SITC meeting intermittent dosing of PD-1 as a possible strategy to examine that effect and see whether we can get reinduction of Ki-67 and T cell proliferation after that first dose. So I think that would be it.
On this side.
So in the context of like monotherapy drug development, I wanted to ask sort of like a challenge question, which will sound funny coming from an immunotherapist, but should it be the case we should actually take fewer molecules into the clinic? And the reason I ask this is if you go back, and so I trained with Mark Ratain, and so he drilled this into my brain a million times, but if you were going to develop a new drug, you would want to know which patient it's going to work in, and you'd want to have some readout that tells you can measure something about the drug changing the biological system that is specific to the drug. And yet I think a summary of all this single agent stuff in immuno-oncology is that we don't know who to treat, and we can't measure anything about whether or not this specifically made the difference. I think the agonistic antibodies are the perfect test case. After a decade of agonistic antibodies, we can't show target engagement in any of the clinical trials. So I wonder if you could speak to that. There's such a rush to bring molecules into clinic, and there's so much preclinical literature that suggests this is really exciting, and yet are we actually wasting a lot of time and money if we haven't done the due diligence to figure that out? What are we going to measure that says we actually are going to show an effect and know which patients we actually need to treat before we go in and spend a decade and billions of dollars on developing all these drugs?
Go ahead.
Yeah, so again, that was sort of my final comment. And is there a mechanism that will allow us to explore that question and, without the need to develop a $10 million antibody to start putting into patients? And are there less rigorous ways that need to be in place to allow us to safely examine what the biological effects of the antibodies are? And so again, the questions of Fc receptor function and the epitope binding that was required. Our OX40 antibody, we were very excited about the fact that it had lost that optimal immunologic dose that we saw both in animal models and with other bispecifics. And so, but in patients, it really did nothing in a dose-dependent fashion. And being able to do that in a quicker fashion and being able to interchange the different Fc receptors to see what exactly we wanted to accomplish would be a nice thing to be able to do. And so I know in Australia, for example, the requirements for IND submission are less rigorous, and that might be a strategy for being able to explore a wider array of different epitopes on the antibodies and Fc receptors to really know what we're doing, I think.
Jason, I think this is one of the major probably points in immuno-oncology drug development is proof of mechanism. We're having issues of proof of mechanism. If you listen to the talks this morning, that's major one of them. proof of mechanism for PD-1 was clear. This is why Suzanne mentioned that we knew who are the patients that we're going to be talking in the first place, and we gave it the upper hand. We're rushing a lot into a clinic without a proof of mechanism. We have general mechanism, it activates T cell. Well, who cares if it activates T cell without knowing the whole system or biologic system around T cell? And this is where we're failing, is because we don't know exactly what they're doing and when to do them, and more importantly, how to combine them.
So sometimes fast is slow for him.
Hi, good morning. Osama Fayyad from the Institute for Experiential AI at Northeastern University. I guess I am still struggling with, and especially from the talks around what didn't work, what are the key takeaways, right? Hussein had one clear one, which is, single factor doesn't work, and his job was not even to talk about failures. But from the other ones, what I would love to tap into is kind of, given the experience you've been through, what would you do different, and what do you recommend to the field in terms of, if you had your wish list, what would you break to get there, and then we can work backwards and figure out, how to change things. Because it feels like, in other fields, and I'm not in this field, learning from failure is much more useful. And it feels like we're not learning enough from the failures here.
You mean conducting 5,635?
We certainly analyzed, I think our budget was something in the $3 to $4 million for each of these trials in terms of analyzing samples and looking at flow cytometry outputs, tumor biopsies. Each biopsy was about $10,000 for the multicolor immunohistochemistry. So that's $20,000 for a pair of samples is what we were charged. And so, those 10 samples, I guess, was $200,000. And I'm not sure that we really came up with anything from that analysis. We did see some patients had upregulation of PD-L1 expression, increases in T cells associated with that, but that was not associated with response to the treatment. So we learned some things, but we didn't learn why it didn't. And again, I think we didn't even know what it was we were going after. We had two hypotheses. Would this antibody either enhance T effector cell proliferation, or would it reduce T regulatory cells? And so just knowing that.
But isn't that why you shouldn't have done it?
Yeah.
So I will also address, Osama's point about success and failure. So yes, we succeeded in first line in melanoma, but we're failing miserably in the resistance setting. And I think as we thought, this entire workshop is titled Resistance. And the truth is we actually haven't even agreed on a definition of what resistance is. And so, we've been talking about SITC this morning. So at SITC, we kind of brought together a committee that tried to define resistance to single agent, and we're about to define resistance to combinations. Just in terms of the clinical phenotype, how do these patients present? And there are different subsets of patients that nothing works for from day one. You do single agent PD-1, you do combination, you go to LAG-3, you go to everything else, nothing works. That's a different clinical phenotype that a patient that has a partial response and gets off treatment for a year and then goes back with recurrence. So even trying to subset the clinical phenotype is a first step in terms of being able to understand the mechanism and then which mechanism we're actually trying to target. Because if you open up 5,600 trials, I bet you about 5,000 of them are in PD-1 resistant disease, and they just enroll all patients with PD-1 resistant disease without taking into account what was the clinical course before they got to that point. So there's a lot that we need to spend trying to understand those aspects as well.
So coming from the small molecule world, the key thing in drug development was knowing in the preclinical setting, what are you targeting and what is the response that you're looking for when you go in vivo? And I do think we have challenges with the availability of preclinical models that are sufficiently predictive. Let me go back over to you.
Scott Littman, UC San Diego. I could be off on this, not my area, but it's a question for Dr. Grupp on the CAR T. It seemed to me early in the development, when we heard about CAR T, the sense was, well, this may work, but there's no way to scale this up for patients. And now we're hearing the same thing on dendritic cell vaccination, for instance. It may work, it's exciting, but no way to scale it up. What are the lessons learned from CAR T scaling up, and your thoughts on scaling up dendritic cell or other types of therapy like that?
So I can briefly comment on that. It's a really, really good point. I think there's no question that the experience in the first now 6, 7, if you include Zynteglo, engineered cell approvals, is that cell manufacturing is a lot harder than you think it will be. And then it's harder in the commercial setting than the academic setting. And then it's harder to scale than it ought to be. And then it's also quite expensive. And despite that, it's all happening, right? And I think, in 2014, when we were seeing amazing responses in leukemia and then lymphoma, we're like, wow, how are we ever going to get this out to a single patient in a drug company setting? And clearly it's doable, but you see from a variety of products, and one of, I won't name any, but that there are challenges in terms of scale. And of course, if we go from blood cancer to something working in a common adult solid tumor, then the scale problem just explodes. But the hope is that what we're learning is that you need to take these academic, very manual intensive, but successful manufacturing processes and automate them. And we see that happening across the field. Presented data on Novartis's short-term manufacturing T-charge process, which is much more automated. And there are others as well. That just happens to be the one that I know about. And so I think that there's a fix to this in terms of doing what drug companies are really great at, which is building these automated and scalable platforms. But are we there yet? No. And if we have to do something that requires 10,000 products a year, we don't have that scale. And of course, we also run into cost issues in terms of what these cell therapies actually cost. So how that applies to dendritic cells, I think that, you run into the same problems in terms of you can make 100 products a year, but making 10,000 products a year is a whole different ballgame.
Yeah, it just seems to me with the incredible advances in technology, if you had a therapy, pick dendritic cell vaccine, that was labor intensive for the initial studies and was producing durable benefit, resistant lung cancer, it's hard to believe that Big Pharma or whoever would develop ways to scale that up. But thanks for your response.
Yeah, scaling is an engineering problem. Look at DNA sequencing. We're doing multiple genomes overnight. Now we're out of time, but you've been waiting a long time, so please go ahead very briefly.
So I'm Donna Payer from Sloan Kettering. I'm a novice immunologist, but I'm a systems biologist. And what I've been listening here is a lot of the times word mechanism. And we talk about mechanism, you zoom in to the one molecule and the second molecule it interacts with, and really, really zoomed in thing. But the thing is that the immune cells, it's an entire cell, it's a system, and it doesn't like to be activated. It has multiple mechanisms to inhibit itself, especially you knock down one, resistance develop because it doesn't like that state. And part of, I think, the problem is that we are, just treating it as a one thing where biology likes a sweet spot. So what would it take to collect more quantitative data that looks at how these things interplay and reach balance in the cell? Because I think, particularly if you want to go into combinatorics, you need a more quantitative model. Now, that's easier to collect in mouse, which Susan has told us, is a really bad system. So what would it take to get quantitative data that really allows us to see how all this interconnects and to develop a dynamical model that can help us predict combinations, doses, and timings?
That's a really terrific point, and Samir is going to explain how we do that in his talk later in the meeting. But you're absolutely right. Immunology defined systems biology, and we need that kind of approach. Thank you for a very lively and engaged discussion. This was a wonderful session. You've earned your break.